Hey everybody, we're going to go ahead and start our transcription and translation lecture. So this is going to be both of them put together, right? So it's going to be relatively lengthy, but I'm going to be honest with you. This is going to be a relatively simple idea or something that you've probably already done plenty of times before in your biology classes or probably a biomed or biotechnology. I'm pretty sure you are familiar with this. So it's going to be more so a review more than anything else. This is kind of giving you the idea, the rundown, what we're going to go do. We're going to talk about our different RNA molecules, what they're all used for, and basically just how transcription works, all the different enzymes that help out with it. Then afterwards, we'll get into translation. Okay, so when we are talking about our RNA, right? So you got two types of genetic information. You got DNA, and you also got RNA, right? Deoxyribose nucleic acid and then ribonucleic acids, right? So we're really focusing on the RNA going forward, and that's all this stuff, right? We've already hit DNA. We already know DNA can be copied. All your genes are right there. Cool. Let's actually get into the RNA bit about this now, okay? Now, RNA comes in three flavors. You have M, which is messenger RNA, T, which is transfer RNA, and finally R, which is ribosomal RNA. And each one of them are basically used for something, a little bit different. Okay. So let's go ahead and take a look at them, right? So your mRNA, your messenger RNA, is used in order to carry the genetic information from the nucleus to the ribosome. Now, this is a little bit of a misconstruing because mRNA can also be used in prokaryotes as well. So what I want you to remember is that prokaryotes, they do not have a nucleus, but they still have DNA. So your prokaryote will still make mRNA, it's just not leaving the nucleus. So you can really just say from the DNA to the ribosome, as opposed to always saying the nucleus. So here is an mRNA molecule right there. And then we also have tRNA right here. So tRNA stands for the transfer RNA. These ones will go and deliver amino acids to the corresponding mRNA sequence. They're going to have this thing called an anticodon that allows them to line up correctly to the codon that would be located over here where our mRNA is. So mRNA is the message. tRNA is how we are getting the amino acids to build the molecule. And then finally, you have rRNA, and the purpose of rRNA is ribosomal RNA. This is basically a skeleton to build your ribosome. So we just throw this rRNA on, we build a protein complex on top of that, and then at the end of it, you get your awesome ribosome, and this is where we're going to go and make our proteins. Okay. Now let's talk about the central dogma when it comes down to life, right? So DNA has the ability to replicate itself, right? During the S phase, right? DNA will replicate itself so that way your one cell will have the correct DNA, replicates itself so that way when the cell splits, right, and copies itself, we'll end up having the second one right there. Cool, everything's all great. Now, what exactly the DNA is, it's all your genes. So the thing is that these genes have lots and lots and lots of information. But the question is, right? The question is, how do I access this information? Because I can't just go right to DNA to the final protein, right? What I need to do is turn it into a workable language, which is this RNA. And then from there, the RNA can create the proteins. Now, this jump from the DNA into RNA, we call it transcription, which is what this topic is going to be talking about. While the following video is going to say, well, once you have your RNA, we will then translate it. In translation, it translates into a protein. So transcription, a way to think about transcription, it means to rewrite. We are going to be rewriting the DNA into a workable language. So then that way we can then take this RNA and then translate it, right? You're translating, like if you're translating a script or translating a language, you're deciphering it to figure out what the heck it is that you're trying to make. And again, all this information comes from this DNA over to here. So transcription is a process in which RNA polymerase will use a single DNA template strand to make mRNA. All right, now take a look. What is the direction of transcription? 5 to 3. A, right? So we're going to be going to the 3 to 5 side, right? Kind of like how, remember when we did this for the DNA modeling, right? It's 3 over to here, and then from there, what's going to happen is I'm going to go this way. So it is being created 5 prime to 3 prime over to there, right? And that's what's happening, right? That's the big shtick, right? Now we have this molecule, this enzyme called RNA polymerase. Very similar to DNA polymerase, right? Remember we did DNA polymerase during DNA replication. RNA polymerase is for transcription. So it literally does the exact same as DNA polymerase, except now we are making RNA instead. Oh man, this is where we're going to have a moment. Gang, I'm going to definitely recommend that you write down these terms because there are multiple different ways. Now, I'm going to let you know something. This, like, when I say that there are certain things that frustrate me in the topics here, right? I don't understand. This is like up there with why does photo system 2 happen before photo system 1? You know what I mean? Like, this is one of those times where I just get frustrated because there's too much language to describe the same gosh darn thing. So I want you to just take a moment and like copy this down and specifically use these words right over here. And we're going to go ahead and label them. So pause the video, and then when you're ready, I'm going to go and add some more stuff to it. Okay, hopefully you got this in there. Let's go describe it. Okay, so it says the template DNA strand may also be called the non-coding strand. Wait a second, your template is non-coding, but you're using it in order to go make the thing. This is backwards as backwards as you can get, in my opinion, all right? And this gets people confused. The template is also called the non-coding. Okay, because you're using the template to build the mRNA sequence. It's also known as the minus strand. It can also be called the antisense strand. You have 4 different ways to describe the same gosh darn thing right here. Like, so frustrating. Okay, let's go ahead and explain why it is that we're using this word right here, okay? The template is going to tell you what mRNA should look like. Okay, so we call it a template because it's supposed to tell you this. We call it the non-coding because really what we're looking at, a way to kind of look at DNA is that the blue is the sense, right? The sense. All right, I'm going to go do this in another color so that way I can kind of like keep track of some stuff, okay? Your sense strand, right? Like it makes sense is your coding strand. What you're trying to do is like, this is what you're trying to get, like this guy up here, this is what you're trying to eventually get translated, right? You're trying to turn this into the protein, right? Again, you have these complementary base pairs, which is why we have these guys stitched onto it so that way I can get my double helix structure down. But your goal is that you're really trying to make this thing right here. Okay, so that's why we call it the sense strand, the coding strand, the non-template strand, which, it's just throws me so off with that. All right, but what happens is that what we're going to go do is this set strand, we're going to go and it's going to look, this should look identical or very close to the mRNA sequence that's going to go and leave. Your only difference is that your thymine should be altered or switched up with uracils because uracil exists in RNA and not thymines. Okay, so and then this is what you're going to go do to get us to our actual amino acids to build the specific protein, right? So from this information up here, right, That gave us our mRNA sequence to get the actual protein structure that we have right here. Now, all this other stuff is just kind of like extra jargon where people can kind of get a little confused. And I'm going to go keep on drawing on this, okay? So this right here, right, the antisense strand, this is the only time that this antisense, this minus, this non-coding strand, right, the template strand only shows up. It's the side of the DNA. I don't really care. Cool, I used it, okay? I used it to make my mRNA sequence. So RNA polymerase is going to attach to this guy over to here, right? And we're going to go this way, building something 5 to 3, right? So we have 5 here, 3 there, right? We're building it 5 to 3. So I got my 5 prime to 3 prime guy right there. And then from there, I need to make sure that I line my tRNA up, the anticodons correctly, which is the purpose of these guys right here. The anticodons make sure that they are complementary to the sequence that I have within my mRNA sequence. So A pairs with U pairs with A, G pairs with C, right? So this anticodon is complementary to that codon right there. U pairs with A, C pairs with G, G pairs with C. These guys are complementary to each other. All right, so this is really what is happening, or like the verbiage that we end up using. I despise the fact that there are 4 ways to describe the same gosh darn thing, but this is something you need to be aware of. Okay, again, it's up there with this. Okay, it's like up there with like the other stuff. I'm like, why is this a thing? I don't know. Apparently somebody just wanted to, get credit for some goofy I don't know. I'm going to be off my soapbox. Okay, anyway. So, the steps, how this actually happens, okay? Transcription is a 3-step process. Initiation, RNA polymerase binds to the DNA strand. So boom, here's RNA polymerase. You're going to go ahead and attach to this promoter region or a start region. Okay, cool. That is all that happens during step number 1. Okay, step number 2, elongation. The mRNA sequence is being synthesized from 5 to 3. 5 to 3, and we read it on the 3 to 5 side, right? So as this is going up, right, we are building this 5 prime to 3 prime going in that direction, right? Perfect. There's going to be a stop sequence over here, which is going to eventually get us to the final part. And that final part is going to be termination. And we terminate, the RNA polymerase unbinds, we're stopped doing that, and then from there, transcription, the mRNA sequence is now done. Now, something different that we're going to go and do is talk a little bit about how we modify this mRNA before we get there. Now, you may have talked about this in your biology classes. I'm not too sure about it, but we need to make sure in eukaryotes, right? This is specifically eukaryotes. Now, keep in mind, where is the DNA in a eukaryote? It's in the nucleus, right? So it is in the nucleus, and then maybe here's like the outside of the cell over here, right? So, and like maybe my ribosome, right? Here's my ribosome here, ribosome. And my ribosome is like, right here, whatever, okay? Big unit, small unit. Okay. Now, what I need to do is modify this in order to get it there effectively. Okay, so my mRNA sequence, I need to add what's called a methyl cap and a poly A tail. And that's what we see over here. So here is the methyl cap right here and the poly A tail. I'm going to be going through those nucleopores that are located on my nucleus. We mentioned them very briefly when we talked about the nucleus back in unit 2, but the mRNA is going to need to squeak through the nucleopore and make its way to the ribosome to eventually be made. All right, so we are going to add the poly A tail. We're going to do this methyl cap, this GTP cap. Now, the other thing that we're going to also do is this thing called introns and extrons and alternative splicing. What the heck does that mean? Okay, imagine, okay, so introns and extrons are, you have extra genetic information, and I can kind of remove them in order to make different proteins out of it. All right, so I have an image right here to show you. So here's my DNA and here's my exons, right? and my introns right there. You can see, so introns, exons, right? The exons want to exit. So this is the stuff that wants to exit the nucleus. The introns want to stay in the nucleus. Okay, so what we're going to go do is alter this so that way I can remove the introns. So I'm going to go and remove the introns to my final guy in here. And what I can do is alter this to make different proteins out of this. All right, I'm going to go do like a little sentence and show you like the alternate display. So let's go take a look, right? So for example, here's my DNA and my RNA. Okay, so here's an exon here, here, and here. Okay, cool. What I'm going to go do is I'm going to remove the introns. It's called RNA editing. And what I'm going to do is I'm going to keep the introns inside, and now I have this mRNA sequence that's nothing but the good stuff. Now, what's cool about alternative splicing is that I can actually, using the same sequence, DNA sequence, I can make different mRNA molecules depending on which exons I keep. So I got exon 1, 2, 3, 4, and 5. So if I go and take a look, protein A has 1, 2, 3, 4, 5. I use them all. But take a look at protein B. Which one did I remove? 3. So there's no 3s. I got 1, 2, 4, and 5 right here. And similarly over here, I got 1, 2, 3, but no 4 in this one. This is alternative splicing. So we're going to go see this in a hemoglobin gene that I have where we can actually see how we can splice it at different parts in order to go and get different proteins out of it. So you're able to get a lot of different proteins, different combinations, I guess, like outcomes from it. All right, think about it like this. Okay, let's say that hypothetically I wanted to go write a sentence, and I'm doing this from, so a fat cat ate the rat, then sat on Pat. All right, perfect. A fat cat ate the rat, then sat on Pat. Really goofy thing, right? But what I can end up doing is like, so my introns and exons, I can end up like changing what the message is going to go be. So maybe let's say hypothetically, fat cat ate and then rat sat Pat, right? These are my exons while the other stuff is my introns. Okay, I can end up doing one combination saying fat rat sat. And that is like, that is one statement. That is one combination of these exons I can make some, right? So the fat rat sat. Or I can be like cat ate Pat. Poor Pat. But no, but you should be able to see it, right? So that's what we're doing. We're just changing, right? Of the exons of what we were going to use, we can basically splice how we want this protein to be formed, and you can get, right, structure determines function. You change up the proteins, you change up the function of the proteins, you're basically able to get a lot more bang from your buck from that. It is pretty neat stuff. So that's that. Okay. Finally, the last thing I was just saying is like putting it all together. Look, this is literally what we just went over, right? So you got your DNA, right? We're going to go ahead and unzip our DNA, RNA polymerase. We're going to go ahead and build my RNA molecule from 5 to 3. Then with my coding strand, right? I'm going to use that to build that coding strand is what I'm trying to make. So then I'm going to go and use the template strand or the non-coding strand to make my RNA molecule to eventually go to the next spot, which is what? Translation. All right, if you need to go take a pause, go take a pause. But we are getting right into this. All right, so translation. We have now made my mRNA sequence. I have now edited it. I have done everything I need to go do. So how is it that I actually make the protein? I'm not going to go into this. We already showed you this. Okay. Where does translation occur? Okay. Where does it occur? There are two places in eukaryotes. Okay. You have ribosomes which happen in eukaryotes and prokaryotes, but also you have the rough endoplasmic reticulum that's in eukaryotic cells only. So these are the two places that protein synthesis can occur. You're going to see all these pictures of these ribbon-looking things going through ribosomes. That's the mRNA. And we're just going to go see the mRNA going through and then the protein being made in the process of it, okay? And we typically show these proteins as like little tiny spheres and balls because that's the easiest way to simplify what these amino acids look like. All right, now something unique about prokaryotes, right? Because prokaryotes do not have a nucleus, they don't do transcription and then do translation later on. They're able to do both at the same time, which is kind of weird to think about. So what you're looking at right here is you are seeing that from the DNA of my prokaryote, here are multiple mRNA sequences, and I'm legitimately building the protein at the exact same time. So the ribosome is building out the proteins and is making all these proteins. This is a electron microscope image of that actually happening. It's really, really cool. So this is why bacteria have such like a short life cycle or a quick binary fission time, because they're just able to be faster at um, replicating things that they need, making proteins that they need in order to survive and reproduce and go about their business, right? Versus eukaryotes is a little bit more specialized, more structured. And again, we keep on saying eukaryotic cells are way more advanced and way more complex than your prokaryotic counterparts. It's not bad to be prokaryotic, but I mean, it's just, it works for them, but they're just stuck at being bacteria at that point. Okay. Really neat thing, right? So remember how it was initiation, elongation, and termination for transcription? Guess what? We use the same exact words for this one too. Oh God, what a time to be alive. Okay. So when it comes down to it, it's the exact same thing. You're going to initiate the translation process. You are going to elongate your amino acid sequence, and then we're going to eventually terminate it. So initiation, we're going to start with what's called a start codon, which is AUG, which always codes for methionine. This is your start codon. So what we're going to go do is get to the start codon. We build the ribosome around it, starting with the smaller subunit, then the larger subunit, and that's what we're seeing right here. Now that we are set here, we are going to be able to go and read each codon, each set of 3 mRNA nucleotides, and then that's going to tell us what we're going to go need. Here is that anticodon that we're going to use to make sure that it pairs up. A pairs with U and C pairs with G, and that's why we have it set up the way that it's set up. Now, elongation, there is a bit of a process that goes along with it. If you have any questions about it, you can go ahead and ask. But elongation is a little bit simple, okay? So here is my methionine right here, okay? Now I get my next tRNA bringing the other amino acid next to it, okay? They end up recognizing each other, right? So like the codons pair up, so the codon and the anticodon pair on up. And then from there, this is going to allow there to be a dehydration synthesis reaction to occur. And now I have attached 2 of my amino acids together. And we're going to consistently move here to the left. And then from there, when we get to this spot, this E spot, this tRNA can go ahead and mess off, go away, go try to collect another amino acid, and now I have 2 amino acids at this point. Now take a look. Okay, I'm going to go change it to purple because I want you to kind of see what's happening, right? So now there is a space at the A spot. So if you got to take a look at this, right? This is where we're at like right now. I can now add another tRNA to here. I'm going to go ahead and attach it to the other amino acid sequence. And what we're basically doing is that we're just building off this amino acid sequence. So you should be able to kind of follow that as it's going further and further and further out, we're consistently elongating this this amino acid sequence. This is why we call it elongation. So these tRNAs are consistently going around and around and around and around and around, delivering their amino acids to grow out the chain more and more and more and more. And keep in mind about your intermolecular forces, right? You have these hydrogen bonds, these hydrophilic interactions, hydrophobic interactions. So this protein is going to be folding in a really, really funky way in order to have its preferred shape, and then it's going to have its particular function associated with it. And then we get to termination, right? Termination is when you hit a stop codon, which is a really unique codon that has no tRNA associated with it. But it's still, we put the mRNA in here. And what happens is that it's just kind of like a placeholder. And then boom, the amino acid sequence ends up falling on off. We break apart the ribosome and we just drive on to the next one. That's all termination is. Now, when it comes down to your amino acids, there are 20 total amino acids with 64 combinations. This is when you end up getting to your codon wheel. Now, you are not going to need to memorize these or know what each one stands for. I personally like the wheel, but the wheel is never showing up onto, never ever ever shows up on the AP exam. It's always the codon chart. The codon chart, I think, is less user friendly because it's easier to mess it up. But what I'm going to go do is go through a couple of examples on how to read it. So let's say AUG, that start codon, right? AUG. So to do it here, I go to the first letter, which is A, which means I am in this row right here. So I have to go here, which here, right? Okay, now I'm going to go to U. So here is U. Second letter is U. So now I go into here, my little box. So now it says I am in this spot right here. And then finally, I go to my last letter, third letter, G. I look at G right here, methionine, right? So A, U, G is for methionine. And if I go and take a look at the codon chart, we do the same thing. We instead now start in the middle. So here we go. We go A here to begin with. All right. Then we go to U. And then you go to G. AUG, there's methionine right there. Okay. Now, where you have some other things is let's say that we did UCC, right? So let's go ahead and reset UCC. Taking a look at it. C. Looks like, let's see here. Sorry. I said UCC. UCC, my bad. UCC, you're at serine, right? UCC, you're at serine. Now, one thing you notice is that there are 20 different amino acids, but 64 combinations, because if you know UCC or UCG, you're still at serine right here. So there's many different codons that are out there that will code for the exact same amino acid. All right. And this is basically more or less the process. Like you're just going to go to the ribosome, right? You have your mRNA, you start it, and then boom, you make your whole entire protein. And then this protein is going to go about doing its business. You make lactase, cool, congratulations. You made an enzyme that's going to go digest milk sugar. You need to make DNA polymerase, cool, congratulations. You made an enzyme that's going to go copy down DNA. You need to make RNA polymerase, cool, congratulations. You now made an enzyme to go and make RNA. Okay, I want to go and make I don't know, a glucose receptor protein. Cool, congratulations. You made a glucose receptor protein that's going to go on the outside of the cell in order to, look for a glucose ligand to start the cell communication process. Like this is how all this stuff happens. Like this is actually what is making all the proteins that we've been kind of discussing and talking about in class. So really, really freaking cool stuff. Okay, so I'm telling you. There's also this other thing about retroviruses. We'll go into detail about it. Basically, with retroviruses, normally I know I've already mentioned this before, but I'll go ahead and do it one more time, right? DNA to RNA to protein. Normally, we end up going this direction. This is the normal central dogma. In the case here, these viruses start with RNA. They go backwards with a thing called reverse transcriptase, an enzyme to make it into DNA, and then we turn it into, we then do transcription to make RNA, and then eventually make more protein to make more virus and stuff. So, it's that. All right, guys, that's that. Thank you so much for listening. We're going to go ahead and have some fun, play some bingo, do this, make sure you're all squared away with it, okay? Take care. See you on the next one.